Okay, so it's time now. Let's start. Hello, everyone. Good evening, and thanks for joining today's webinar on scalable microfluidizer technology for cosmetic applications. I'm Daljit Singh, Microfluidics Sales Director for India and Southeast Asia. And on behalf of Microfluidics team and our local channel partners, I extend a very warm welcome to all our guests in the today's webinar. Thank you for joining and we appreciate your time. As you all know, our today's speaker is Dr. Yang Su. Just a brief about our speaker. Dr. Yang is currently Manager of New Technology and Application at Microfluidics International Corporation based at our Boston office. He has very rich experience working on R&D optimization, processes, and application development across a variety of industries. So great to have Dr. Yang with us today to share some of his knowledge and expertise in cosmetic applications. Few housekeeping stuff for convenience of everyone. All participant cameras should be turned off and microphones muted. If you have a question, please submit it through the chat window at bottom right corner of the screen, and we'll have it responded in the Q&A session after the presentation. Q&A session will be handled by my co-host, Matthew Baumer. Please note this webinar is recorded and will be hosted on Microfluidics global website and used for promotional activities. With that, I hand it over to Dr. Yang for the presentation. All yours, Dr. Yang.
Thank you, David. And hi, everyone. Thanks everyone again for joining today's webinar. As Daoji mentioned, the title today suggested that I will be introducing you the scalable microfluidizer technology, and hopefully use some examples to demonstrate how this technology can be applied for cosmetic applications. Here's a brief agenda for today's presentation. I'll be starting talking you a little bit more about Microfluidics as a company, and then switch to explain how the technology works. Before I move on to talk about more case studies, I want to briefly discuss about some of the considerations you should consider when you're selecting your manufacturing technologies or method, and why uniform processing are important and what the benefit it can provide. Then I'll move on to talk a bit more detailed examples for using the microfluidizer technology for the cosmetic industry. The example includes solvent-free perfume, specialty shampoo, liposomal formulation, and an interesting topic around self-disruption for cosmetic application. Then I'll give you a quick example, illustrate how the technology is able to scale up linearly. Then I'll show you, briefly introduce you our diverse product portfolio before I end my presentation. So who's Microfluidics? Microfluidics has been around for more than 40 years, and we specialize in design and producing high-pure fluid processors using our unique in-action chamber technology. We're located in Massachusetts, just outside of Boston, but we have a global presence, have been sold many processors to our customers all over the world. Back in 2011, Microfluidics joined, which is now our parent company, IDEX, and grouped with a few other companies to form the material processing technology platform, or the MPT platform. And our technology is very versatile and can be used for many different industries, including pharmaceutical, cosmetics, nutraceutical, as well as chemical industries for various different type of applications and products. And we have systems covers from development all the way to manufacturing. And with our vast applications and machine design experience, we are able to tailor our design and solution to meet each one of our customers' requirements, because we think we believe our customer success is our success. So this is a quick summary of some of the applications that the technology works very well. It's ranging from liquid-liquid systems, including emulsions, liposomes, polymer nanoparticles, and lipid nanoparticles, all the way to dispersions such as cell disruptions, different deagglomeration, particle size reduction, which are all good applications for our technology. And as I mentioned, we have systems that covers the entire R&D all the way to pilot and to production, which are all using our unique interaction chamber designs. Now, this diagram here illustrates how the system works. Samples with large particles enter the system via the inlet reservoir and is pressurized by a constant pressure intensifier pump. So the pressure can go as high as 30,000 psi. And at such high pressure, the product is pushed through the fixed geometry interaction chamber where a microchannel resides in there, and the product experiences uniform high shear treatment to achieve particle size reduction. After processing through the interaction chamber, the product temperature can be efficiently controlled by a cooling device such as cooling coil or heat exchanger, depending on the machine, to be efficiently control the product temperature if needed. We have two different interaction chamber designs. The one shown on the top here is what we call the white type chamber. You can see the white chamber has two microchannels. Sample come into the chamber and will be split into two streams. Each will pass through the microchannel subjecting to the high shear treatment and then impinge on each other after exiting the channel. So this design is more suitable for dispersing liquid-liquid samples, which includes nanoemulsions, liposomes, lipid nanoparticles, and polymer nanoparticles, etc. The design shown at the bottom here is the Z-type chamber. The Z chamber only has one microchannel. The sample will go through the channel, subjecting to the high shear treatment and impact on the solid wall at the outlet. So this design is more ideal for solid containing samples. So it can be used for various different dispersions, including cell disruption. One of the big advantages of the microfluidizer technology is the combination of fixed geometry interaction chamber and a constant pressure intensifier pump, which can achieve constant pressure. This is different from typical or conventional valve-based high-pressure homogenizers. So I have a cartoon here that illustrates the effect of the homogenizer pump on the valve. So the homogenizer basically usually based on the constant volume. So the pump is constantly pumping the same volume through the valve. And the valve is designed with a base and the valve and the base. So the pump keeps pushing through, it will generate tiny vibrations to the valve because the gap is usually controlled by a spring. So that tiny vibration is exaggerated here in this animation here, but if you look at the pressure profile, which is shown here, you can see actually that tiny vibration can translate into as a huge pressure variation, which showed in the red curve here as the actual pressure measured for a high pressure homogenizer. If your set pressure is 30,000 psi in this case, you can see the pressure variation can be as high as 50%. So you can drop the pressure as low as 15,000 psi here, which means your material is not receiving a uniform treatment. It is being treated as a variety of different pressures, which will not result in a very efficient process and uniform product. Whereas in the case of microfluidizer technology, as I mentioned before, we were able to achieve a constant pressure by combination of the chamber and the pump design. You can see as shown in the green curve here, after a quick initial acceleration of the intensifier pump, where pressure ramps up, you can reach to the set pressure and maintain that pressure during the majority of your cycle, and then your material will receive a very uniform processing at the same pressure there. So after at the finish of the cycle, the intensifier pump retracts and get ready for the next one. So that's why the pressure drops to 0, but you don't need to worry because nothing get processed in this period. And the curve shows below here is a translate from the pressure to shear rate. So that's exactly what I mean by earlier by uniform treatment. You can see the variation of the shear rate and versus the constant shear rate from the microfluidizer technology. So another big advantage of the microfluidizer technology is our guaranteed linear scalability. So when we scale up, we don't increase or enlarge our microchannel, as if you, in the case of conventional homogenizer, you usually have to use a bigger gap to allow higher throughputs. So what we do is we actually keep the microchannel dimensions, but we would use a parallel line of multiple channels. So by using this type of design, we will ensure as long as you keep your processing condition the same, especially the processing pressure, the materials will receive exact high shear treatment inside each microchannel, and hence to achieve guaranteed scale up results. Now, before I move on to talk about applications, I just want to quickly use this slide to discuss some of the considerations that when you choose your manufacturing process and develop your product and processes. So I would say, obviously, the most important thing are the target properties, especially the physical chemical properties. When we're talking about nanomaterials and nanotechnologies, obviously, subparticle size and size distribution, I think it's probably the most important thing. At least one of the most important things, because you need to be able to achieve certain size and maintain that good size or narrow size distribution. And often when you achieve certain size and distribution, it will also lead to desired properties for the product, such as viscosity, texture, and etc. Now, when you look for a method to produce or process your sample, obviously you want your method of selection to be very repeatable, that can consistently generate batch results. And the method should also be somewhat future-proof when you're ready to scale up. The method should be ready to scale up directly for larger scale manufacturing. Some of the other considerations that listed here may not specifically apply to cosmetic industry or applications, but it may as well be. So the selection of the manufacturing process should also allow easier downstream processes. I have one example here, which is often used for pharmaceutical applications, meaning a lot of the product, especially the parental product, needs to be sterilized, and sterile filter is typically used to help remove bacteria. And the filter has an average pore size of 0.22 micron or 220 nanometers. Obviously, that indicates the majority of the particles in your product or in the parental drugs should be less than that to achieve a good filtration efficiency or to allow an efficient filtration process. And last but not least is a lot of the manufacturing, depending on the industry you are, is subject to different regulatory requirements. For pharmaceutical, again, using pharmaceutical industry as an example, the process needs to be compliant with the CGMP requirements. So the selection of the equipment should allow the whole process to be able to comply with those requirements. I've mentioned when I introduced the technology, I mentioned a lot about uniform processing that you can achieve constant pressure and constant shear rate. So as a benefit of the uniform processing, I mentioned about when I talk about particle size and distribution is you're not only able to achieve a desired particle size quicker, but you're also going to get very narrow distribution. So the benefit of ROSE you can certainly achieve better product stability. Better stability often translate to or mean a prolonged shelf life. So the increased shelf life of the product is always a good. And I also mentioned about texture, viscosity stuff. So the particle size and distribution can also allow you to achieve desired or even better appearance and feel of the product. And the last thing is, if you are able to achieve a very efficient process, that means you are able to save a lot on the production costs, such as you can save energy using less number of passes and etc. Now, move on to talk a little bit more about cosmetic applications. I listed here a few different formulations that are commonly used for cosmetic industry or the cosmetic products. I think the first formulation are emulsions. Everyone probably know this very well, that emulsions are widely used in cosmetic products, including shampoo, perfume, lotion, cream, all kinds of different products. Liposome is also another commonly used system that can use to help encapsulate and deliver functional cosmetic ingredients. And there are various other formulations that are based on dispersions, such as facial body exfoliating scrubs, lipsticks, temporary tattoo ink, and collagen. And as I mentioned before, I listed as a separate category for self-disruption, which is a very interesting application. I will talk a little bit more later. So out of all these different formulations, you don't always need to use a high pressure treatment or advanced technology to make your product. It is needed and important when you need to specifically produce high value added and often utilizing nanomaterials for your product or your applications. That's where you need a robust technology to achieve that. So the drive for using more and more into the nanotechnologies and nanomaterials is the customers shift to focus more on health and appearance of the product or of themselves, and hence they demand the product to include more functional cosmetic ingredients and efficient delivery systems. So to produce nanomaterials, again, particle size is very important. I've talked about that already. So you want to precisely control the particle size because when you talk about nano, it's not just a value, it's a range of the particle size. So you can be as little as add a few nanometers to maybe 100 nanometers, or to several 100 nanometers, or even slightly bigger to close to sub-micron particle size. So each different ingredients and product will probably have requirement for different particle size. So you want your technology to be able to help you precisely deal in that condition and achieve the size. And achieve desired particle size also helps you to achieve better nutrient fragrance or active encapsulation, and in return achieve targeted or enhanced transdermal delivery of those actives that's being encapsulated in the formulation. And I've mentioned about appearance and physical properties already. And often use of nanotechnologies and nanomaterials with precisely controlled particle size can allow you to create innovative formulations or products that use fewer additives or undesired chemicals or solvents to help your product be more natural, which is a trend that consumers are looking for. And by utilizing those approaches above I mentioned, so you can get a lot return in your investment. I've mentioned a few already, with such as increased shelf life. You can do continuous production, and waste less materials. You can save on the production cost. You can potentially simplify your downstream processing, and most importantly, your process is ready to scale up. You don't have to waste more time and energy to redevelop or reinvent your process on a larger scale. With that, I move on to my first example, which is nanoemulsions. So just a quick background slide here in case you're not familiar with nanoemulsions. So the definition of emulsion is two or more immiscible fluids with one dispersed in the other or others. So it's important to note that the nanoemulsion is not same as microemulsion. In the case of nanoemulsion, they're not thermodynamically stable, but they're kinetically stable, which means there's a different stability mechanisms that can affect the stability for the nanoemulsions. One of which is the so-called Alstor ripening phenomenon, where you have large droplets, especially around or bigger than 1 micron in the size, can slowly absorb the smaller droplets and the larger droplets will become bigger and bigger and eventually will lead to separation or some kind of instability for the product or the emulsions. Emulsions are widely used not just for cosmetic industry, pretty much you can see it everywhere in any other industries. And depending on which is the dispersed phase or the continuous phase, you can think about the droplets and the continuous phase. You can either form a water oil emulsion or oil and water emulsion, or even multiple emulsions. You can have double emulsions or even more. And because oil and water are immiscible, so you do need some sort of surfactant or emulsifiers, or some people call stabilizers, to help maintain the droplets from coalesce with each other. That's how you maintain the droplets and the stability of the emulsion. I would say by far, oil and water emulsion is probably the most commonly used because of their ability to carry and deliver hydrophobic actives. A lot of actives, especially functional cosmetic ingredients, are water insoluble, so you would need a carrier oil to achieve encapsulation and delivery of those ingredients. So this leads to the first example of making an emulsion for alcohol-free perfume. That I think everyone knows that alcohol is often the main ingredient for perfume formulations because it is used as a solvent for the aromatic compounds, which are mostly water insoluble. There are some drawbacks when alcohol is present in the product because it can cause skin dehydration and skin barrier damage, especially for sensitive skins. So as I mentioned before, there's a new trend that push to remove solvent from the formulation. This is also applicable for perfume. Consumers are looking for cleaner product and greener product, or more natural product. So the perfume companies are now working on to develop water-based alcohol-free formulations. As I mentioned before, emulsions, which is oil and water emulsion, is probably the best approach because you can select the carrier oil to help carry the aromatic compounds and make them into the perfume product. Some of the challenges is you want to hopefully with alternative formulation, but still able to maintain or resemble the alcohol-based product. Especially one of the property is the appearance. A lot of the perfume product are translucent, so you certainly want to somehow mimic that appearance with your emulsion formulation. So to achieve that, you definitely need to control and drive the particle size because the smaller emulsion droplet size is certainly going to help in this in this case to help the the visual appearance of the product. So we worked with one of the leading cosmetic companies to help them process the formulation they developed for emulsion to encapsulate fragrance. And as I mentioned before, to be able to help to achieve a desired appearance of the product, final product. In this case here, we were looking to reduce the particle size greatly to around about 100 nanometers. As you can see the results here, that the unprocessed means the coarse emulsion before processing through the microfluidizer processor. The size of the emulsion, if you look at the mean particle size, it is just a little less than 10 microns. As you can also see from the particle size distribution curves here, where you have very large particles to begin with. But as soon as you process through the microfluidizer processor, even just after one pass, you can see the particle size was reduced dramatically with a mean particle size less than 200 nanometers, as indicated by this curve here. And every additional passes through the system will further reduce the size of this emulsion. And after 5 passes, we were able to successfully achieve the goal in this study here with the mean particle size just slightly over 100 nanometers, as shown in this green curve here. You can see the beautiful shift from the large particles to the smaller particles after processing. The second study or second case study for emulsion is it's a formulation developed for a specialty shampoo. So this shampoo was developed for bond treatment because bonds are very important because they're responsible for healthier hair. When bonds are broken, it can lead to brittle, damaged, and eventually broken hair spreads. So this is not a regular daily shampoo. It's a specialty shampoo. That means you need to have your functional ingredients there. You need to achieve a certain desired particle size. In this case, we want to produce a stable, homogeneous emulsion with a D50 particle size less than 200 nanometers. If you're not familiar, D50 is a term used in particle size analysis that representing 50% of the population or the droplets are less than a certain size. In this case, it's 200 nanometers. And D90 with a similar analogy is 90% of the droplets to less than 300 nanometers. And again, you can see the results showing both in this table here and the distribution curve here. Again, we have very large droplets of the emulsion to begin with, the D50, which is close to mean particle size in this case. It is a little over 5 microns. You can see the wide distribution of the coarse emulsion, and in this case, after just one pass through our system, we were able to reduce the D50 particle size to less than 150 nanometers, and the D90 particle size was less than 200 nanometers, which is a lot smaller than the target in this process. And the efficiency of the technology allowed our customer to only process one pass to be able to achieve the particle size, which actually helps a lot in this case. Is you don't have to repeat the processes, and with the high efficiency means you can achieve a higher throughput, and which in turn will help in your production of the actual product. Now move on to a different formulation, which is liposome. In case you're not familiar with liposome, again, we're going to just briefly introduce liposomes here. Liposomes are artificial spherical lipid vesicles with a bilayered structure. Phospholipids are usually the main ingredients to form liposomes. There are usually other different type of lipid ingredients, but phospholipids is probably the the most commonly used. And the phospholipid has a hydrophilic head and a hydrophobic fatty acid tail. That which means when you disperse them in water, because of the hydrophobic tail, they will actually self-assemble into the bilayered vesicles with the tails facing each other, and the hydrophilic heads are facing either outward and inward. So that means there's also an aqueous core being encapsulated inside the capsules. That makes the liposome a very versatile delivery system, and it's a very popular and very successful system that are commonly used for a lot of different cosmetic products. With the different head and tail groups, it is capable of encapsulating either hydrophobic, which will be inside the bilayer region, or the hydrophilic, which will be inside the aqueous core, or both at the same time. Some examples include different peptides, vitamins, and CoQ10. With the bilayer structure, liposome vesicles can effectively protect the encapsulated actives from undesired effects from external conditions, and they can enhance penetration through skin barriers, which lead to better thermal bioavailability. Because of the aqueous core they encapsulate, liposome can stay on the surface and to help increase skin hydration. Last but not least, they can help protect skin from external stressors by forming an occlusive layer. So here, showing you a quick example of using the liposome formulation to encapsulate and deliver botulinum toxin. People probably know this very well, that this is a highly potent neurotoxic protein. But in cosmetic applications, it is used for reduction of facial wrinkles by relaxing the facial muscles. So everyone probably heard Botox already, which is utilizing this highly effective ingredient. So here we're trying to encapsulate this with the liposome formulation. Obviously, to achieve a desired transdermal delivery, we wanted to reduce the particle size of the liposome formulation to below 200 nanometers in this case. Again, you can see before pressing through our system, the D90 particle size is somewhere around 10 micron in the size. You can actually see that with the microscope image showed here on the top. You can see all the large vesicles. And after passing through the microfluidizer processor, the D90 particle size, again, meaning 90% of the vesicles or populations are below 150 nanometers in this case. And from the microscope image, you barely can see anything. The reason being the resolution of the microscope is not as high that you can be able to see nano-sized particles. Anything less than a micron, it won't be able to show up here because you can see this scale bar here represent 20 microns. Now I'm going to switch gear a little bit here to talk about a different application, which is cell disruption for cosmetic applications. I found this is very interesting. The concept of using cells to or microbes to produce important products such as enzymes, proteins, and other products are not new. People have been doing that for more than 60 years. If you think cells, they can be they can be viewed as a little factory, a living factory that can, potentially produce and grow a lot of different functional ingredients within the cell. A lot of some of the very common cells are used, whether for bio industry or for cosmetic industry, are bacterial cells and yeast cells. So for bacterial cells, E. coli is one of the most commonly used, I would say. And then for yeast cells and a typical baker's yeast or brewer's yeast is also very widely used. And so the reason that you would need cell rupture is not the cells will not always, depending on the type of the cell and the product you want to harvest from the cells, they are not always being expressed extracellularly, meaning that cell will not excrete the product from within the cell. So in that case, the product of interest are encapsulated or captured inside the cell that you need to rupture the cell, either to help release the intracellular content, or if it's something such as a membrane protein, you have to rupture the cells and collect the cell membrane debris and do further extraction and purification of your product. So same thing applied to cosmetic industry. You can use cells to grow a variety of different functional cosmetic ingredients. And often you will need to perform cell lysing process to help harvest the product of interest from the cells. And also by doing this is biosynthetic is viewed as a natural product process, not same as chemical synthetic process. So the product or the ingredients developed through this process can be viewed as a natural ingredients or source of natural ingredients. The last thing I want to mention here is there's an increased interest or trend that uses fermented cosmetic that derived from microorganisms. This is studied from I think somewhere early 2000 and have been growing very fast. It's originated from Japan and Korea, and now has grown into the second largest cosmetic group after the herbal cosmetic group. So the fermented cosmetic ingredients are, again, naturally derived. They're more potent, can provide better skin penetration, help support skin barrier, and reduce irritation and inflammation. So they're often considered as innovative and product and have competitive advantage on the market. Again, to fully utilize the potential of this fermented cosmetic ingredients, you often have to perform cell lysing process and use a cell lysate as one of the ingredients for your product. So I mentioned the importance of rupturing the cell to release the functional ingredients or the product from within the cell. And I also mentioned there's a variety of different type of cells that can be used. Each will actually is different and will require different shear forces to break open the cell membrane. So for example, mammalian cells are usually pretty easy to rupture, and they're on the low end, and bacterial cells usually in the medium high shear rate ranges, and yeast cells are often tougher, will require probably the highest shear forces to rupture them. So depending on what type of cells and products you're working and interested in, you would need a technology or a method that can cover a wide range of the shear rate, and so you can efficiently break the cells. And the important thing is you need to precisely pinpoint and dial in this shear rate. So what you want is the just enough shear rate to open or rupture the cells, but not too much so you can potentially degrade the product or damage the product of interest. So that's where Microfluidizer technology comes into play. As I mentioned before, we can precisely control the shear rate by selection of processing pressure and the induction chamber, because we're able to produce uniform processing. So we can use that to precisely open or lyse the cells and not to damage the product of interest. The example I'm going to talk about here is bacterial cell. Again, as I mentioned before, bacterial cell is very commonly used, and they can produce many different products. The interest cell here is the Micrococcus luteus. So Micrococcus luteus is a gram-positive spherical shaped bacterium, hence the name of coccus. representing the spherical shape. So they're among the skin-dwelling microorganisms, which means they are found by human skin as well as many other places. So they have a lot of potentials for promoting skin health. Micrococcus luteus exhibit antimicrobial activities against a wide range of microbes, which makes them a potential candidate for topical probiotic applications. They also have inhibitory activities against certain pathogenic microbes, which can be potentially used for against infections. So they're also capable of producing DNA repair enzymes, which will help repair skins and revitalizing the skins. And one other important or interesting property of this bacteria is they can produce pigments that within them, which allow them to withstand massive dose of UV radiation, which means they're not being used as active ingredients in many different cosmetic products, but they can also be used for products such as sunscreens. Again, example here illustrate how efficiently we can use the microfluidizer technology to perform cell lysing for this cells, and you can see before processing. The microscope image here show you all the intact cells. Because the size of the cell are small, you can see a lot of the cell density is pretty high here, but you can see them through the microscope image, and they're also confirmed by doing the particle size analysis. Although in this case, the microscope is probably the better analysis tool. However, the particle size can also indicate the results before and after a performed cell lysing step. So before, you can see the larger particle size with the distribution here. They're in the micron range. Some of the larger size may indicate the agglomeration of the cells. And after just simply passing through the microfluidizer processor once, you can see the reduction, dramatic reduction in the particle size. Most of the particle size were now within even less than a micron, which is showing on this huge peak here. That means the generation of very small cell debris. And from the microscope image here, you can also see barely see any intact cells anymore. Again, due to the resolution, you will not be able to see the small cell fragments, but at least the image shows you that the efficiency of the process that leaves no intact cells behind. Most of the cells have been ruptured. Okay, my last example is a scale-up example. Hope to use this example to demonstrate the scalability of the microfluidizer technology. So here the goal is pretty straightforward, that to scale up an emulsion formulation that was developed for a cosmetic product. So the formulation and the process was developed using a benchtop microfluidizer processor, which in this case was a LM20 microfluidizer processor. Showing here, so you can see again the efficiency of the process that reduces the particle size to the desired size very efficiently. You have the large particles before processing through the system, and after processing through the microfluidizer processor LM20, we were able to achieve an average particle size at around 200 nanometers very efficiently with a very narrow distribution, as indicated by this peak here. And then we're ready to scale this process up. So as I mentioned before, when we scale up, we use the multi-slotted induction chamber design. In this case, we use this chamber on our larger production scale machine, which is a M7250 model. So this machine is capable of delivering up to 5 liters per minute at the maximum pressure of 30,000 psi. So you can see the results here when we process the same formulation through there's two different systems under exact same condition. We were able to achieve almost identical processing results. You can see the average particle size very similar, around 200 nanometers, with a good polydispersity index. The PDI indicate the distribution of the particle size. And from the distribution curve here, you can see they're all almost overlapping on top of each other that indicate the excellence, the scalability of the technology. So you can directly scale up from your benchtop R&D development to the production scale. So that's all my examples for today. I want to use this slide to quickly show you our product portfolio. On the top row here, or top line here, shows all of our benchtop units. We have different benchtop unit. They come as a slightly different specifications, but they're all capable of processing a variety of different formulations. And the bottom row here shows you our small pilot scale unit, large pilot, small production unit, and all the way to our larger production system. So most of the pilot and production system are highly customizable. We have a lot of different options that can suit your unique processing requirements. With that, I'd like to thank everyone again for your attention today, and be happy to answer any questions that you may have.
Thanks, Dr. Yang, for such an insightful presentation. If you have a question, please type it in the chat window, and we'll respond it.
Okay, Daljit, we have some questions come in already during the session. So the first one is how the microfluidics can be used in industrial large-scale production.
Yeah, so like I said, we have design of different production scale machines. So the design is primarily to ensure we have a pumping system that is capable of deliver higher throughput. They will be using our same in-action chamber technology. So everything you do or you have done with the lab scale processes can scale directly. We will choose the same in-action chamber design. We will determine the number of the microchannels inside to be able to meet the capacity of the system. So basically, you will just to be able to ready to process a larger batch. Although there's a few considerations usually when you scale up, and but I think we can discuss in more detail offline if needed.
Okay, thank you. So the next question that I have is there a viscosity limitation to using your tools?
I would say that's a good question. Well, the quick answer is yes, at some point. In general, the system and the technology is able to handle a wide range of viscosity. We have seen some very high viscosity formulations. The pump is powerful enough to pump higher viscosity samples or product through the in-action chamber, the microchannel inside there, without any issue usually. But usually, I think the issue with the viscosity is how you transfer the product from from your inlet, either the reservoir or tank, to the system and collect it. And so on the larger system, we use we would pair the machine with a feed pump. The feed pump is powerful enough to help you feed the machine. On the benchtop unit, usually we will rely on gravity fed. So there often the limit of the viscosity is hopefully the sample is still flowable and pourable. So that's our general rule of thumb. But we do have some tools designed to help if viscosity is an issue. So I would say in general, we would process something more as a liquid, but I mentioned we have done different lotions and creams with our systems.
Okay, thank you. So the next question I had was, is temperature control possible?
Yeah, the short answer is yes. It can be done differently from the smaller system to the larger system. On benchtop unit, we have usually we have a cooling device. What we call we use the cooling coil. You can use ice water bath if you need to cool the temperature, or you can use the warm bath to maintain a warm temperature if needed. Some of the units on the lab scale also have options for using heat exchanger, which is a better temperature controllability. You can utilize either a chiller or recirculating a different cooling medium to help control the temperature. On the larger systems, all of our systems have the cooling capability. On the larger system, is paired with heat exchangers, so give you option to control, and for certain systems, we have options actually providing a capability of closed loop temperature control, where we'll put a temperature probe to measure product temperature, and you can the system is going to use that to regulate the cooling flow rate to achieve a desired or maintain the desired product temperature.
Okay, thank you. And I just note that if anyone wants to follow up with either of the speakers, you can do that via the website, via the contact us form, or you can contact them at their emails on screen. I've also dropped those into the chat for people that might find them useful. I've got another question, please. Which parameters do you need to control to obtain a uniform particle size distribution? And how can you assess the uniformity?
So I would say the uniform particle size is somewhat inherent from the technology and the machine design. Like I said, our constant pressure intensifier pump with the fixed geometry induction chamber will achieve constant process pressure, hence will give you the uniform particle size and processing. However, there are some parameters you can use to achieve a desired particle size. Pressure is one of them, and is one of the important parameter, because it will affect the shear rate produced in the microchannel. We also have, as I mentioned, we also have different type of induction chambers that can be used for different applications. And the dimension of the microchannels are also another parameter that we have different designs that has different channel dimensions for different induction chambers. So with the combination of that, we would be able to pinpoint the desired shear rate that produces the desired particle size. And there are a few other parameters can be considered during the process. For a lot of emulsion, I would say probably also true for cosmetic emulsions, temperature is important. Someone just asked about temperature control. Then another thing I mentioned there is the number of passes. Sometimes you can achieve your final particle size within one pass. Sometimes you may have to repeat the process when we say running for multiple passes. So that's all common. That's a few parameters. But like I said, the parameters will help you achieve the particle size, and the uniformity will come with our the advantage of the technology. So I would say the best way to assess the uniformity is by measuring particle size. You can see with all my examples, I was able to show you the particle size and distribution. So the width of the curve and some particle size analysis method will give you a number to indicate the uniformity of your sample. So I'd say that's probably the best way.
Thank you. There's a few moments left. We've got time for a few more questions. We will also type up responses to these questions and make them available on the Microfluidics blog. The next question is, can the microchannel withstand such large pressure considering the viscous flow?
Yes. So our system, including the microchannel, is designed to withstand that pressure. They're rated more than the achievable maximum pressure of 30,000 psi. So all the high pressure components, including the induction chamber, yes. And I didn't mention the core of the microchannel. We even have options to use a very robust material, which is diamond. So we have diamond version of our induction chamber that offers excellent, wear resistance to give you a long processing life.
Okay, thank you. Next question. In the presentation, emulsions and liposomes were discussed. Is this applicable for solid lipid nanoparticles?
Yeah, certainly. The way using our technology to create solid lipid nanoparticles is actually somewhat it's done somewhat similar to creating the emulsion. Obviously, the system cannot process the solid lipid by themselves. So usually you would have to heat up your formulation above the melting point of the lipids. So then the liquid become a liquid form, and then you can disperse that in that aqueous phase, then running through our system, just like you are processing emulsion. So after processing, you will achieve very small particle size of your lipids, and then you can, cool the product, reduce the temperature, allow the lipid to solidify, become tiny or nano-sized lipid particles.
Okay, thank you. We've probably got time for one more question. For achieving water-based emulsions, i.e. oil and water emulsion, the requirement of the surfactants is necessary, correct? Will
Yes,
that
right.
be a disadvantage?
If I understand correct, is the disadvantage associated with the water and oil emulsion specifically, or with the surfactant? I'm not quite sure, but I would say in general, the system can also be used, or technology can be used to create water and oil emulsion, but just inherent difference between water and oil and water emulsion is oil and water emulsion is a lot more stable, I would say, than the water and oil emulsion, because due to the surface tension, water usually tend to be a continuous phase. So you do have to work on the formulation to adjust the ratio of the water and the oil, and also you choose a better surfactant. So I would say formulation is very important, and then work on that. You can build on that with the fine tuning the processing conditions to make a water and oil emulsion.
Okay, thank you. We've got one last question. I know we just hit our time. So, how does Microfluidics encapsulate active ingredient into liposomes?
Yeah, so good question. So there's a different ways. It's mostly associated how you form the liposome. I didn't have time to explain that more, but if you remember, I showed the formation of the vesicles. If you have a lipophilic ingredients, you typically would want to premix that with your lipids or the mixture of lipids. Then when you are forming liposomes by dispersing the lipids in the aqueous phase, lipids usually self-assemble into vesicles. But the issue is when they self-assemble, they form very large vesicles, sometimes giant vesicles, or multiple layered vesicles that will actually impact the efficiency of loading or encapsulation of the active ingredients. So our system comes into further pressing the hydrated lipid vesicles to help peel down the layer and reduce the size to form small unilaminar vesicles. But in through this process, the lipophilic ingredients are already embedded with the lipids. So that's how you encapsulate them. For hydrophobic. So for hydrophilic ingredients, they will be usually dissolved in the aqueous phase. So when you're forming liposome vesicles, they will be encapsulated in the aqueous core. So that's how you encapsulate them.
Okay, thank you. There are no more questions for the time being. Just as a reminder, yeah, please
Matt,
contact the team. Yes.
Matt, we have another question that comes to me.
Okay.
Dr. Yang, that is regarding the scale up of our processor. So the question is, when you use multiple micro channels parallelly, how do you ensure the flow behavior, such as the pressure and the flow rate in different channels, are the same?
Right, great question. So we do a lot of work into our design process to design our multiple channel chambers. The one thing we need to make sure, like I mentioned in the presentation, is you need to be able to maintain the pressure. Obviously, that's what I said earlier. When we use a multi-slotted interaction chamber for a larger scale machine, we will carefully choose the number of the channels so the machine can still achieve the desired process pressure. So maintain that. So as long as the machine is capable of maintaining that pressure, we know the pressure across each channel will be the same and the material will receive the same treatment. And this has been demonstrated and proven over and over by many of our customers using our production system, including one of the example I've showed in my presentation.
Okay.
Thank you.
Thank you, Dr. Yang and Matt. So appreciate all the questions from our participants, and thank you very much for your active participation. However, in the interest of the time, we have to wrap up the event now. If you have any question following this session or require any application specific support, please feel free to get in touch with us directly. Emails provided on the screen or contact your local channel partner. So with that, it is time to wrap up the event now. We hope that you found the today's session informative and to be a valuable learning experience. We would like to express our sincere gratitude to all of you for attending this webinar. Thank you very much for joining and have a great evening.
Thanks everyone again.
Bye.
Thanks, Daoji. Thanks, Matt.
Thanks.